Imagine a world where drug discovery happens quickly and accurately. At Topogen, we focus on finding new topo drugs, essential in the cancer fight. Our mission includes understanding how these drugs work in both normal and cancer cells to create life-saving treatments. Topo-targeting drugs generally fall into two categories. First are the interfacial poisons or IFP agents. These drugs arrest the enzyme in a DNA-bound state, leading to DNA damage and cell death. These drugs essentially redirect cellular topoenzymes into DNA-damaging agents that cleave the genome. Second are the catalytic inhibitory compounds or CIC drugs. These agents block enzymatic activity without forming stable DNA drug complexes and offer a different therapeutic inroad. Since topoenzymes are essential for many aspects of DNA metabolism, blocking their activity can prevent the cell from functioning normally, leading to cell death. Our high-precision screening assays can differentiate between these two modes of action, providing critical insights into drug function. For this video, we compare IFP drug screening for both TOP1 and TOP2 enzymes. Let's discuss more specifics about interfacial poisons. IFP action is similar between the two types of enzymes. However, because they interact differently with DNA, the poisoning event is quite distinctive. Looking first at TOP1, the basic enzymatic function involves nicking and resealing of one strand of DNA. This will generate a relaxed form of DNA derived from a superhelical DNA substrate. Single-strand nicks are transient and always followed by an immediate resealing step. Therefore, single-strand nicks are not detected since the breakage and rejoining reaction is tightly coupled. This protects the genome from random breaks arising from unregulated TOP1 action. The IFP drug binds at the transient cleavage site to block the resealing step, making the cleavage permanent. To detect cleavages, one simply reacts TOP1 and DNA followed by treating with SDS to denature the active enzyme. This freezes the cleavage complex, yielding a stable DNA topo complex. The nicked DNA complex is measured using an agarose gel containing ethidium bromide. The situation for TOP2 is quite different. The TOP2 reaction intermediate is a double-strand break, and in this case the IFP drug will generate linear DNA, as shown in the lower diagram. Without IFP drugs, neither TOP1 nor TOP2 causes lasting DNA breaks. This explains why topo drugs are highly effective anti-cancer agents. IFP drugs subvert a beneficial and essential topo enzyme into a DNA damage complex. We show results for a leading IFP mechanism using a kDNA substrate. The results display a decatenation test with the IFP drug VP16. First, let's look at the markers. Lane 1 on the left side shows that the kDNA is a high molecular weight complex of interlocked 2.6 kilobase rings that cannot enter the gel due to its large size. A linear kDNA marker is also shown. The next lane shows a typical TOP2 reaction with kDNA. TOP2 unlinks the kDNA mini-circles from the network by transient double-strand DNA breaks, releasing two mini-circular DNA bands shown here. The upper is nicked mini-circular DNA and the lower is circular DNA lacking nicks. The next lane shows an IFP drug result. The red circle is linear kDNA, which forms because of IFP action of VP16 and TOP2. In the absence of IFP drugs, linear kDNA not detected. The basis for the kDNA assay with TOP2 is described here. A typical assay result is shown in the gel below, and as you can see, there is no linear kDNA formation in the absence of an IFP. In this experiment, we demonstrate how to identify a TOP1 IFP using plasmid relaxation. We use an EB-containing gel to spot the TOP1 cleavage product, which is nicked open circular DNA. We always include markers in lanes 1 and 2 as they are crucial for data interpretation. Lane 3 serves as an IFP control with CPT, showing the formation of nicked DNA. Lane 5 is a negative control without the test drug. Lanes 5 to 9 contain increasing amounts of drug X, highlighting the accumulation of the nicked DNA. This indicates that the test drug is a potent IFP agent. The lower gel shows how we detect TOP1 catalytic activity in a non-EB gel format. Supercoiled and linear DNA markers are visible. The IFP nicked product is indicated with a red arrow. Minus and plus CPT lanes show relaxation products as a variety of DNA bands with different linking numbers. Note that the nicked DNA cleavage product is hard to resolve in gels without EB. Stated differently, non-EB gels are better for observing catalytic TOP1 activity, but less effective for measuring the nicked IFP product. For instance, Compare the separation of nicked DNA in lane 3 in the EB gel to lane 4 in the non-EB gel. Clearly, the EB gel is optimal for detecting nicked DNA, while the non-EB gel is better for assessing relaxation or TOP1's catalytic activity. Using these tests, we can understand complex drug interactions, leading to new drug discoveries. If your lab is new to these experiments, we can handle the analysis for you. It's fast and affordable. We offer complete solutions with experienced managers. Contact us for a quote.